homozygotes. Homozygote. Or sometimes we can call it pure breeding. Homozygotes. Sometimes we can actually call it pure breeding. The term we must use, the term here simply means pair. That's pair two of character that are similar. Homozygotes are pair of character, pair of pure breeding, pair of similar alleles or character or traits. That is homozygotes. But okay, look at the example of homozygotes. Just like the example we used to describe hybrid, I can describe this as homozygotes because the pair, the two genes are similar. So this is a clear case of homozygotes. This could be used to connote a tall gene, that in terms of physical appearance. We can have a case where we use AA. Most time I can use this AA since it is pair. The two genes are the same. They are pair of similar traits. So this can connote skin color, or I can use it as normal skin color. It is pure breeding or homozygotes, similar pair. We can use, but whenever we use similar pairs and they are both represented in capital letter, like you can see TT, AA, you can see BB, and they are all in uppercase letter. This type of homozygote or pure breeding are called dominants. I can call this homozygote dominant. Homozygote dominant. That is if this pair of similar character are represented in uppercase or capital letter. The other aspect of homozygote is homozygote recessive. That is if we have opposite, maybe if this is written as contrastive pair of small letter t, so this has become recessive, and this can be written as this. This has become recessive, and this can be also written like this, has also become recessive. But for the fact that they are pair of similar character, similar allele, so this can be regarded as homozygotes recessive based on their symbols. These ones are represented symbolically in capital letter and this in a small letter. But both of them are called pure breeding. Pure breeding, this one will be pure breeding in the aspects of dominance. Pure breeding in the aspect of recessive. But if you look at the third terms we use here is heterozygotes. Now, heterozygote is an outcome of homozygotes. If I want to obtain a heterozygote, I must have a homozygote. Okay, if I want to obtain a heterozygote, it must be a cross between a homozygote dominant and homozygote recessive. So the outcome of these two gives heterozygotes. Will it be heterozygote dominant or heterozygote recessive? Of course, it should be heterozygote dominant. Okay, let's look at this example. Supposing we have this dominant normal skin color. Let's use this as dominant for normal skin color, and let's use this for allele for albino. People who lack that special pigment in the skin tend to have this type of gene. Now, supposing I want to see the outcome of a cross between these two, and then I have the parental. When I say parental phenotype, it means that the physical appearance of the parents were one normal skin color, versus albinism. This is what's the physical appearance of the two parents connotes. And then if we want to look at the genotype now, if we want to look at the genotype now, since normal skin is the dominant trait, we connote it with capital or uppercase letter AA. And then we are crossing with albinism. Albino gene will be represented by lowercase letter. Okay, now in genetics, there must be cell division of which describes meiosis. These pairs of gametes will segregate or separate or disjunct during meiosis, one from each parent. It's not going to be in pair again, but each will be singly inherited. Now we're going to have A instead of AA. This A will step aside this way. We have this, and then this. And then this is what is contained in the sex gametes. And then in genetic cross, this happens during what we call random fertilization, whereby this at randomly unite with this to give me this, and then this randomly unite with this to give me this. And I have this with this, I have this, I have this. I can call this F1 offspring. That is, the first offspring of this cross is called F1. If you look at the F1 offspring, since we have earlier said that these uppercase letters or capital letters are dominant and the lowercase letters are recessive, and when this dominant character are crossed with the recessive character, all the offspring will appear to look phenotypically like the dominant character. So what it means that all offspring here will have normal skin color, irrespective of this abnormal skin color of albinism. That means this offspring will not show any clear case of albinism, even though they have albino traits. They are carrier of albino, but for the fact that this normal skin is dominant, the dominant character will always predominate the recessive character. So this will all appear to have normal skin. But when they have that normal skin, both of them are both homozygotes. But this is homozygote dominant, and then we have homozygote recessive. But this is neither homozygote recessive or homozygote dominant. But this case where we have two unsimilar pair, one uppercase and the other lowercase. Since the two are unsimilar, this describes a condition of heterozygosity. And you can no longer describe this as pure breeding again, but you can interchangeably use the word impure breeding. That is opposite of pure is now impure. If the pair of characters are unsimilar, it is heterozygote. I repeat, if the pair of characters are unsimilar, it describes heterozygote. If the pair are unsimilar, it is heterozygote. If the pair are unsimilar, it is heterozygote or impure breeding. Just like the example I gave, TT, AA, and then BB. And then the other contrasting pair could be the lower cases, AA, and then BB. These two can go and give me an impure of this. This can go and give me an impure of this. And then we have this with this. I can have this impure. So these are impure breeding or heterozygous Heterozygotes. Heterozygotes. So, heterozygotes are a pair of unsimilar character. Pair of unsimilar character are heterozygote, or we refer to them as impure breeding. And then we have another term we use in genetics, which is hemizygotes. People usually interchange the two, but they are not the same. You cannot say either hemi or hetero. No, hemi is quite different from heterozygotes. Heterozygote is when the pair are not similar. Hemizygote is also when the pair are not similar. But there is one common feature that is different between hetero and hemi. If hetero are pair of unsimilar, hemi are pair of unsimilar, or in an example of hemi, one of the examples of hemi we can use human blood group. Human blood group, that is human AB blood group, and then XY chromosome in human. The two pair of AB are unsimilar. Just like this is unsimilar, this is unsimilar, this is unsimilar too. But we cannot use heterozygote to describe this or this, because we cannot obtain any of this by mating or matching homozygote dominant and homozygote recessive. The outcome of heterozygotes is the outcome of a cross between a homozygote dominant and homozygote recessive. We give you any of these and we describe them as impure or heterozygote. But the case of 8b is not a cross between homozygote dominant and recessive. No, it is not. And this is why this is not described as hetero, but they are hemi. Both A and B are dominant. They are codominant. This is not the case where you have B and small b, where B is dominant, capital is dominant, this is recessive, and therefore everything here we go towards the dominant character. That's not the case with this. Now, this is dominant as well as this is dominant. So no one here is more dominant than the other. And because no one here is more dominant than the other, we describe this as hemi instead of heterozygote. Example of hemizygote can be seen in what? Human AB and the XY chromosome, sex chromosome in human. And then we look at alleles.